talking about the tongue, okay. So, main functions of the tongue. The tongue, of course, plays a really important role in speech. In the context of the digestive system, the tongue plays an important role in forming the food bolus. So, as food is taken in by mouth, it is mixed with saliva. Chemical digestion of carbohydrates begins. The food is mechanically digested, and then the tongue, using the what we call the papillae, which house taste receptors, are able to push the mechanically digested food mixed with saliva up against the palate, the corrugated portion of the hard palate, and form it into a food bolus so that that bolus can be easily swallowed. Other important functions of the tongue include, of course, taste. So everybody's familiar with what their tongue looks like. And when you look at your tongue, you notice that it has a bunch of bumps on it. Those bumps are what we call papillae. So the papillae are, they're the bumps, if you will, or they're the structures that contain, and not all of them do, I should say, but many of them contain what we call taste receptors. So we have the papillae themselves come in kind of four general categories based on shape and to some degree location as well. So at the front, the most anterior aspect of the tongue is covered with what we call filiform papillae. Some interesting details about filiform papillae. Filiform papillae oftentimes do not contain taste receptors. They play an important role in providing friction so that that food bolus can be easily formed. Sometimes some of them will have taste receptors, but many of them do not. We also have what are called fungiform papillae, which are called fungiform because they're roughly mushroom shaped. You kind of find those typically right in the middle of your tongue. Circumvallate are the larger, as you can see in this picture, the larger papillae that tend to be back toward the posterior aspect of the tongue. And then the foliate papillae you find, as you can see here, typically on the medial and lateral aspects of the tongue. And the fungiform, circumvallate, and foliate all, for the most part, contain taste receptors. Okay, for salivary glands, so saliva is made up, the fluid saliva is made up of two different types of secretions. It's made up of what we call mucousy secretions and what we call serous secretions. Serous secretions are the ones that are more watery in nature, and then mucousy secretions are, of course, much more viscous because they have a larger degree of mucus in them. The secretions themselves come from the three different salivary glands that we find. So the salivary glands that we have, as you can see in this picture, are the parotid gland. Then we have what are called the submandibular gland. And then we have what's called the sublingual gland. The parotid gland is the largest of the three, and its secretions are mostly serous in nature, almost entirely serous, so the watery type of saliva. The sublingual gland, which you can see here under the tongue, those secretions are mostly mucus secretions. And then the submandibular gland, its secretions are a mix of serous and mucus. Saliva itself plays a really important role in cleansing the mouth. So we have within the saliva, we have enzymes such as lysozyme. We have IgA, as we know, is secretory immunoglobulin that helps protect against specific antigen, and then various other defensins and chemical components that help to cleanse the mouth and keep it, obviously, we're not talking about a sterile environment, but help to keep it cleansed. Yeah, I'm trying to think of a more articulate way to put it, but think of it as cleansed. Saliva plays such an important role, in fact, in keeping the mouth cleansed, that if for example, somebody has to take a medication and one of the side effects is having extreme dry mouth, and this goes on for a period of time, that makes an individual very susceptible to developing what we call dental caries, which are cavities. And ultimately, that having a very dry mouth also makes people susceptible to developing periodontal disease, which I'll show you pictures of in a moment, and you don't want if you can avoid getting it. Another important characteristic of saliva is that it contains salivary amylase, right? And amylases are the enzymes that catalyze the reactions by which carbohydrates are broken down. So what this tells you is that the chemical digestion of carbohydrates begins in the mouth. Okay, so that's what this slide goes over. Are those three different extrinsic salivary glands I made mention of? The parotid, which is the largest, secreting the serous, or the saliva, it secretes serous in nature. Submandibular secretes a mixture of serous and mucus, and then sublingual under the tongue is primarily mucus secretions. And then the intrinsic, to differentiate extrinsic from intrinsic salivary glands, the intrinsic salivary glands are the salivary glands that secrete specifically an enzyme called lingual lipase. So here's the deal with fat digestion in the mouth. If you were to be asked on an exam, unless I specifically asked you about lingual lipase, there's not a great degree of fat digestion that occurs in the mouth. We do have an enzyme, lingual lipase, secreted by the intrinsic salivary glands, that does begin the process by which the chemical bonds in fats are broken. However, the vast majority of lipid digestion occurs in the duodenum. And that is largely because within the mouth, we don't have any bile. So there's no way to emulsify the fats that are exposed to the lingual lipase. So the lingual lipase just kind of gets things going with fats, but it's not, like I said, if you were to be asked on an exam, where does fat digestion occur? Your answer would be in the small intestine. Okay, so just a real quick overview of a homeostatic imbalance that's related to the salivary gland. So mumps is a viral infection of the parotid gland, mostly the parotid gland. The symptoms associated with mumps are, of course, the swollen parotid glands. That's how it gets its name, mumps. It gave people kind of a lumpy appearance in the face. Mumps itself, complications associated with having this viral infection include pancreatitis, encephalitis, and meningitis, which oftentimes are lethal, and also what we call orchitis, which can lead also to permanent hearing loss. So before the vaccine for mumps existed, oftentimes individuals did not leave childhood unscathed. And if they ended up with this infection, could either end up with an infection of the central nervous system, which is what meningitis and encephalitis are, or if they were able to just survive their episode with mumps, they oftentimes were left either sterile. If this person was an adult and a female adult who was pregnant, mumps is oftentimes results in miscarriage because the developing embryo or fetus becomes infected, and then permanent hearing loss is a relatively common side effect of infection with mumps. Okay, so when it comes to teeth, functionally speaking, are there for mechanical digestion. We have what are called, and most people are familiar with the fact that we have baby teeth, and then we have what we call adult teeth, right? The scientific term for baby teeth is deciduous teeth. We being humans have 20 of them. They show up between 3 to 6 months of age, typically. And then those deciduous teeth continue to erupt all the way past the age of 2. And then we have those deciduous teeth until right around the age of 6, when the roots start to be resorbed and those teeth begin to fall out. And those teeth are replaced by 32 what we call permanent teeth. So that's the quote unquote scientific term for adult teeth. Most adult teeth are there by the time an individual reaches the age of 14. And that the exception of that, of course, is the wisdom teeth, the third molars, and those show up oftentimes not until an individual is in their 20s, if they show up at all, because so often these days they are removed. As far as the different categories of teeth we have, in the front we have the incisors, right? You have, here you can see the incisors here. These are the teeth that are kind of flattened, if you will, and are particularly good at gripping and tearing. Behind the incisors, we have what are called canines, which are designed for ripping flesh, or that's what you would speculate based on the fact that other species that are closely related to us. That's what they use those canines for, and structure determines function. And so, of course, it would make sense that canines are involved in tearing flesh, or tearing in general, I should say, maybe not flesh, but tearing. And then we have premolars and in the very, very back molars. And those teeth are particularly well suited for grinding. As far as the different parts of the tooth are concerned, um, I'm not going to ask you too much detail on an exam, but this is what I do want you to know about teeth. So the outer portion of a tooth is covered in what we call enamel. Enamel is the hardest material in the body. So this is a very, very hard substance. And then underneath the enamel, we have what makes up the bulk of the tooth by volume is what we call dentin. Dentin is a darker color than enamel, right? Enamel gives teeth its white color, and dentin is kind of a light brown color. And dentin itself is relatively porous, so it's not very hard and it's not very well protected, which is why if your enamel is eroded due to the acidic secretions of bacteria that have colonized there, for example, and you end up with a cavity, you go to your dentist and the dentist drills out that area and fills it in with a material that substitutes for the enamel. Because if you don't do that, this dentin would quickly erode away, and right underneath the dentin is what we call the pulp. So the pulp is where you find all the nerves and the arteries and the veins. If an infection gets down into the pulp, not only is it incredibly painful, but that infection can very easily travel and get into the circulatory system. And because of the location of the teeth, that infection could easily travel to the brain, which would be highly problematic. And then the last thing I'll say is a root canal. So there are some people who have had root canals. I'm one of them. What happens with the root canal is you have a cavity that was not detected. Essentially, the cavity has gone all the way down into the dentin and down here into the root area. And so in order to ensure that that infection is removed and cannot get into circulation, a periodontist typically, or an I'm sorry, an endodontist, I think actually, goes in, and maybe even some dentists, I'm not sure, go in and they drill out the root, and they pack this with cement, so that there are no more nerves and there's no more vasculature in that area if you have a root canal. Okay, so I'll just go through this pretty quickly. Some homeostatic imbalances that are related to the teeth and to the gums. So the gums themselves are made up of connective tissue, and that connective tissue is largely made up of collagen, which is a really great type of fiber for binding. And the function of the gums is to hold the teeth in place. So dental caries or cavities, I just kind of briefly touched on a moment ago. What I want you to know about them is that they are due to bacteria that have colonized on a tooth, and then the secretions from that bacteria, which are low in pH, end up dissolving through the enamel. And again, then that infection can spread down into the dentin and into the pulp and the root of the tooth, worst case scenario. Gingivitis is it's due to gingivitis is an inflammation of the gums that is typically due to bacteria that have colonized right where the tooth meets up with the gum. And the colonization of that bacteria triggers an immune response. And so inflammation occurs. And in response to that inflammation, the gums start to recede. So some characteristics of gingivitis is that if caught and treated, gingivitis is reversible, meaning that if gingivitis is treated, there doesn't necessarily have to be a loss of gum tissue. And it's treated by removing the calculus, is what we call the name of the colony of bacteria. So when you go get your teeth cleaned, that's what they're doing. They're scraping away the plaque on your tooth and any place that those bacteria have colonized, and hoping to prevent that inflammation from occurring, or to resolve the inflammation that's occurring at the gum line. If gingivitis is not resolved, you can end up with what is called periodontal disease. So periodontal disease is characterized by irreparable tissue loss. So you get recession of the gums that will not grow back, so to speak, or that tissue does not proliferate. and replenish itself. During periodontitis, what happens is there's so much inflammation that you get essentially the immune system activating osteoclasts. So in addition to this loss of gum tissue, which again, the gums hold the teeth in place, you also osteoclasts, just by way of reminder from 227, those are the cells that reabsorb bone. So you end up with bone loss. So the combination of the activation of the osteoclasts and the loss of gum tissue can result in tooth loss. Okay, so the pharynx. The pharynx is the throat, right? We talked about the pharynx in pretty good detail when we were in the respiratory system. There are 3 sections of the pharynx: the nasopharynx, oropharynx, and laryngopharynx. The nasopharynx is a passageway for air only, but food and water go through the oropharynx and the laryngopharynx on their way to the esophagus. Oh, I thought I had one more slide. Okay, well, that's all I have to say right now for this slide. We'll pull this all together when we talk about swallowing here in just a moment. Okay, and then the esophagus, characteristics of the esophagus that I want you to know. So this is a muscular tube that connects the mouth to the stomach. It's a passageway for food and water to the stomach, to go from the mouth to the stomach. The esophagus itself is lined with non-keratinized stratified squamous epithelial tissue, which is a type of tissue that is really well suited to deal with abrasion, because a food bolus is oftentimes causes abrasion as it's making its way down to the stomach. And then once we get into the stomach, we get this transition from non-keratinized stratified squamous epithelial tissue to simple columnar in the stomach, which is a type of tissue that's really well suited for essentially chemical digestion and some absorption. Another important feature of the esophagus, as far as the structures that we find there are concerned, is that within the esophagus, we have globulate cells that play an important role in secreting mucus that coats the bolus and makes it easier to slide down the esophagus on its way to the stomach. Okay, so we've talked about ingestion, which of course occurs at the mouth. Mechanical digestion, mastication is a fancy word for chewing. So mastication occurs in the mouth, right? That's mechanical digestion. We already talked about how we get digestion of carbohydrates starting in the mouth by way of salivary amylase, right? And a teeny bit of chemical fat digestion by way of lingual lipase, but just a teeny bit. And then now we're going to move on and talk about swallowing, which is called deglutition, right? Which is the propulsion of the food bolus from the mouth through the oropharynx and the laryngopharynx, through the esophagus and down into the stomach. So we break up the process of deglutition, which is swallowing, into 3 phases. All right. The first is a voluntary, and so the first phase we call the voluntary phase because it's voluntary, meaning it's under conscious control, right? This is when you put something in your mouth, when you consciously swallow it and move it back toward, and then ultimately through the oropharynx. That's the voluntary phase. The other 2 phases, the pharyngeal phase and the esophageal phase, are not voluntary. They're involuntary. The pharyngeal phase involves reflexes that are, or I should say, involves sensory input that is relayed to the medulla oblongata, and a reflex that's involved with pushing that food bolus down through ultimately down into the esophagus. And then the esophageal phase, which starts when that bolus makes its way into the esophagus. And we're going to break these down to more detail right here. So what I want you to know about the voluntary phase is obviously it's voluntary, and as a result, it's controlled by the cerebral cortex, right? The frontal lobe. You make a decision to swallow, you take a sip of water, you eat a bite of food, and you consciously decide to swallow it. The pharyngeal phase is the first of the involuntary phases. It's initiated by sensory receptors, tactile sensory receptors. And then that sensory information is taken to the medulla oblongata in an area called the swallowing center. Other important characteristics of the pharyngeal phase is that the signals that are sent to the medulla oblongata, those signals and part of that reflex ensures that you are not able to breathe and swallow during the same time, right? Because And if that reflex is disrupted, that's when you choke on food, right? You don't want to be able to, the epiglottis, as we discussed when we were in the respiratory system, is the structure that, as we are swallowing something, prevents food and water, I should say, from going down into the trachea. And that is ultimately controlled by the medulla oblongata. And then the esophageal phase, also involuntary. This starts when that bolus of food that was swallowed passes through the esophagus. Having the presence of the food bolus in the esophagus stimulates peristalsis and helps to propel that bolus down toward the stomach. And then what happens, another reflex that's associated with the esophageal phase, is the opening of the sphincters. So the esophageal sphincters that typically remain closed because, as we'll talk about when we get to the stomach here in just a second, the stomach is very acidic, right? Because there's so much hydrochloric acid that is secreted within the stomach. And so usually the esophageal sphincter is kept closed to prevent hydrochloric acid from splashing into the esophagus. However, as part of the reflex during the esophageal phase, that sphincter relaxes, which allows the passageway of the bolus from the esophagus. And again, peristalsis is propelling that bolus along and pushes it down into the stomach. Okay, I'm not going to spend a lot of time on this, but there are some homeostatic imbalances that are related to the esophagus, to the esophagus and the esophageal sphincter. And two examples of those would be what we call gastroesophageal reflux disease, or GERD, commonly known as heartburn. And then in extreme cases, not extreme cases, but I guess kind of GERD gone wrong can result in hiatal hernia, or the two are kind of tied. Well, give me a second, I'll describe what I'm trying to say. So GERD itself is usually due to relaxation of the esophageal sphincter. And that relaxation causes the stomach, or allows for the stomach contents to enter into the esophagus. And this is problematic. It causes heartburn. That's the term used to describe it because it's uncomfortable. But in addition to being uncomfortable, this phenomenon, having chronic GERD, can predispose an individual to developing esophageal cancer. And that's because exposure of that non-keratinized stratified squamous epithelial tissue that we find in the esophagus to various low pH acidic stomach contents causes metaplasia to occur. That's what it's called, Barrett's esophagus. And you get this transition from stratified squamous epithelial tissue to simple columnar epithelial tissue, which is the kind of tissue that we find in the stomach that is able to handle the low pH of the stomach contents really well, but is not very well suited for dealing with abrasion. And so that metaplastic event is considered a kind of a precancerous event. And then a hiatal hernias occur when the stomach starts to get pushed up toward the esophagus. So this can occur, there's relaxation of the ligaments, the structures that hold the stomach in place as we age. Also, for women who have been pregnant, there's the process of pregnancy displaces all of the internal organs, and the way in which the stomach is displaced is up toward the esophagus. And so pregnant women are not pregnant women, and then even women after giving birth are particularly susceptible later on in life to developing a hiatal hernia. But the problem with the hiatal hernia, or when it can become really problematic, is if the stomach completely invaginates into the esophagus. And that can, in fact, choke an individual. So in that case, the stomach is essentially pulled out of the esophagus and then stapled down or anchored in place. Okay, so now we are going to talk about the physiology. What happens? Well, we're going to start talking about gross and net anatomical features of the stomach, and then we'll talk about the physiological processes that occur in the stomach. So when we look at the stomach, right, some of this is lab related, but I do expect you to at least, in order to, again, structure determines function. So in order to understand what happens in the stomach, you have to have an appreciation for the anatomical features and landmarks of the stomach. So in this image here, you can see here's where the esophagus connects in to the stomach. This area right here is called the cardiac region, or the cardia, right? And we have what's called the cardiac sphincter, which is that's the cardiac sphincter. When I was saying esophageal sphincter, there's esophageal sphincters that are volant, that are present kind of right above, and then anyway, suffice it to say, you can kind of use cardiac sphincter to describe the esophageal sphincter. It's a sphincter that controls passageway of the bolus from the esophagus into the stomach. But this is the cardiac, where's my cursor? This is the cardiac region. Then the top of the stomach here is what we call the fundus. This is the body. And then the last portion, the most inferior portion of the stomach is what we call the pyloric region, or the pylorus. And then the sphincter that connects the stomach to the small intestine, the first portion of the small intestine, which is the duodenum, is what we call the pyloric sphincter. Some things to keep in mind when you're looking at this image is that when we were talking about tunics, we talked about the outer longitudinal layer and then the inner circular layer, right? In addition to those layers, we have a third layer in the stomach, a third muscular layer of the stomach. And that is really important because the stomach, the main function of the stomach, in addition to, I should say, chemical digestion, is what we call mixing, taking the bolus of food that was just ingested and just entered the stomach, mixing it with the secretions in the stomach, and then churning that all around to both mechanically and chemically digest that food stuff, and turn it into what we call chyme, which is a liquid material that is very acidic because of all of the hydrochloric acid that is present. So that is what this slide goes over as far as the layers are concerned. Again, just remember, within the stomach, there are 3 layers of smooth muscle, not 2, which is what we find in the tunics of all of the other areas of the alimentary canal. When we look at the mucosa of the stomach, you'll see that we have what are called gastric pits. This is a histological specimen that lead into gastric glands. Those gastric glands are important for producing the secretions that are used to make chyme, to both chemically and mechanically digest the food. And again, simple columnar epithelial tissue is the type of tissue that lines the stomach. Okay, so those glands, what are the cell types that make up those glands? So we have what are called surface mucus cells. Surface mucus cells are found kind of at the top portion of the gland primarily. They secrete an alkaline type of mucus, right? Mucin is the name that we use to describe mucus before it's been hydrated. So this is just a precursor to mucus. So surface mucus cells secrete alkaline mucus, which helps to protect the epithelial tissue of the stomach from erosion. Then we also have what are called mucus neck cells, which also secrete mucus. They secrete an acidic form of mucus. Parietal cells, so parietal cells are the cells that secrete both intrinsic factor and hydrochloric acid, or they secrete the components that will come together to make hydrochloric acid. More on that in just a moment. And then chief cells secrete what's called pepsinogen. So pepsinogen is the precursor to pepsin. Pepsin is an enzyme that is responsible for digesting proteins, and pepsinogen gets converted to pepsin in the presence of hydrochloric acid. You need hydrochloric acid to convert pepsinogen to pepsin, or to do it with any degree of efficiency. If hydrochloric acid, not enough hydrochloric acid is around, pepsinogen does not get converted very efficiently to pepsin, and that makes protein digestion much harder than it otherwise would be. The other secretion that chief cells are responsible for are gastric lipase. So again, we had some lingual lipase in the mouth that started the digestion of fats. We have some gastric lipase in the stomach that continues a little bit of digestion of fat, but as you'll see when we get to the small intestine, the vast majority of fat digestion occurs in the duodenum, because that's where bile is dumped, and bile emulsifies fat, which makes lipases work much, much better at digesting lipids. And then very importantly, we have G cells. So G cells are what we call enteroendocrine cells. The word endocrine tells you that we're talking about a cell that secretes hormones. So these are the cells that secrete gastrin, right? Gastrin is a hormone, so of course it gets secreted into the blood. And it was the hormone that I told you on Monday to think about as, you know, it's kind of a silly way to think of it. Think of it as the pro-stomach hormone. And by pro, I mean gastrin is a stomach that initiates and promotes activity within the stomach. It increases activity in the stomach. Okay, So here's a picture that shows you how parietal cells are responsible for making hydrochloric acid, right? So what the parietal cells do, this is showing you blood right here, and then this is essentially the gland that's going to lead up to the lumen of the stomach, and here's the parietal cell. So what happens is that water that's made available to the parietal cell is split, and I mean spontaneously dissociates into a hydrogen ion, where's my cursor? Hydrogen ion and a hydroxyl group, right? Carbon dioxide, which is a byproduct of cellular metabolism, combines with the hydroxyl group to form bicarb. And then what happens, and this is very important, this bicarb leaves the parietal cell and goes into the blood, and the bicarb itself is an anion, right? So it has a negative charge. So in order to keep charges consistent, as bicarb leaves the parietal cell and goes into the blood, chloride from the blood is exchanged for that bicarb. So chloride enters the parietal cell and then quickly diffuses out here into the lumen of the stomach, right? And then remember this hydrogen ion I brought up when I was talking about the spontaneous dissociation of water? Well, this hydrogen ion leaves through, there's embedded in the membrane of the parietal cells are hydrogen potassium pumps. And so the hydrogen that is produced is pumped from the parietal cell into the lumen of the stomach. And when you get hydrogen coming together, hydrogen ion coming together with the chloride ion, they come together to form hydrochloric acid. So that is how the parietal cell makes hydrochloric acid. Intrinsic factor, the reason intrinsic factor is important. So intrinsic factor is required for vitamin B12 to be absorbed within the small intestine. Drawing back into when we were talking about the blood and we were talking about pernicious anemia, right? So that's a type of anemia in which individuals do not, their parietal cells aren't functioning correctly, in the sense that not enough intrinsic factor, if any at all, is being produced. And those individuals are very susceptible to becoming anemic. And the reason they become anemic is because vitamin B12 plays a really important role in the development of erythrocytes. And so if you do not have enough vitamin B12 in your blood, the red blood cells that you make are not are somewhat defective. So they don't do their job very well, as an easy way to think of it. And again, that is why intrinsic factor plays a role in the development of that anemia if it's not present. And then chief cell secretions, I already mentioned that pepsinogen is converted to pepsin by hydrochloric acid. And then again, the enteroendocrine cells secrete gastrin, and the gastrin plays a really important role in essentially stimulating all of the activities that occur in the stomach. So gastrin will stimulate stomach motility, increase contractions, that mixing that's occurring in the stomach, increase smooth muscle contractions at the stomach. Gastrin also is stimulating to parietal cells, so it increases the amount of hydrochloric acid that is produced. It is also stimulating to chief cells. So you end up, in the presence of gastrin, you get increased amount of secretion within the stomach, and you get increased motility. Okay, so I'm not going to spend a lot of time on this side. I just want to say gastritis is the general term used to describe inflammation, inflammation associated with the stomach. Gastritis can lead to the development of what we call peptic ulcers, which is erosion of the stomach wall. Many of these ulcers are caused by a bacteria called H. pylori. We don't have time to spend on it, but it's interesting if people want to go look it up. It was probably around 2000 and I don't know, 5, maybe not even that early, but I'm going to say 2005, that this association was there was enough empirical evidence to conclusively determine that H. pylori is in fact associated with the development of peptic ulcers. That is all I'm going to say about that. And then moving on to part 2. Okay, so now we're going to talk about activity within the stomach, right? So we've already discussed mixing. Mixing is made possible by the fact that we have the 3 layers of smooth muscle that we find in the stomach. And mixing is a very important form of mechanical digestion. The stomach is the contractions produced by the stomach are pretty powerful. And as I already mentioned, what they do, those contractions do, is they mix whatever was taken in by mouth with the secretions of the stomach, and in doing so, they produce chyme, which has a very low pH because of the hydrochloric acid. And then what happens is gastric emptying. So gastric emptying happens when there's enough pressure in the stomach that pressure, and we'll talk about contents within the stomach more in just a moment. But as the stomach is mixing up the contents themselves are moving down, being kind of sloshed around and moving down into the pyloric region. So as the pressure in the pyloric region increases, that opens up the pyloric sphincter, which is what controls the movement of substances from the stomach to the duodenum. And the chyme is I'm trying to find another word aside from squirt. The chyme is moved into the duodenum in small volumes, a couple of mils at a time. And then what happens is it gets moved, gets squirted, for lack of a better way to describe it, into the duodenum. And then there's a little bit, as the sphincter closes, there's a little bit of what we call retropulsion, which is the movement of that chyme back into the stomach. And that just further helps to churn up the contents of the stomach as they are moving from the stomach into the small intestine. Okay, so some other important characteristics of the stomach. The stomach, I mentioned this on Monday, but the stomach, kind of like, or much like the cardiac system, the stomach has its own way of the tissue itself. The muscles can depolarize spontaneously, just like what we see with nodal cells. That spontaneous depolarization helps to ensure that the alimentary canal is always contracting and moving, that it's always moving to some degree. As far as the force of contraction is mostly controlled by the extrinsic controls, so the parasympathetic and the sympathetic nervous system, in addition to gastric gland secretion. So gastric gland secretion is mostly controlled by parasympathetic and sympathetic innervation. And when we talk about the phases of contraction and what occurs within the stomach, we break the events, and I should say in the stomach and between the stomach and the small intestine, we break those phases, or we organize them into 3 phases: cephalic, the gastric, and the intestinal. So the cephalic phase is a phase that is initiated by the thought of food. What happens when you think of food is you have signals that are sent to the hypothalamus, and essentially what that does is it increases vagal stimulation, which is parasympathetic nervous system stimulation of the stomach, and the stomach starts to increase its production of secretions. So the parasympathetic nervous system stimulates those enteroendocrine cells in the stomach to secrete gastrin, stimulates the parietal cells to start making hydrochloric acid, stimulates the chief cells to start secreting pepsinogen, and it kind of preps the stomach. It's the phase that is the anticipatory phase for food. So it preps the stomach to receive food. When you're hungry and your stomach is growling, that's because the cephalic phase has been initiated. So the stomach starts to move because of the presence of gastrin, and it's getting ready to receive food, which it does in the gastric phase. So the gastric phase is regulated through a reflex called the gastric reflex, and also through the release of gastrin, right? So the reflex itself, the gastric reflex, is initiated by stretch receptors. We call those receptors baroreceptors, right? We've talked about baroreceptors before. We also have chemoreceptors in the stomach that are specifically there to detect the presence of protein. So when you ingest a meal that is high in protein, that protein acts as a buffer to the hydrochloric acid that is present, and the pH of the stomach increases, right? That increase in pH is detected by chemoreceptors. So that increased pH is indicative to the stomach that that protein is present, and as such, it'll stimulate the chief cells to start to start secreting more pepsinogen, which in the presence of hydrochloric acid gets converted to pepsin, so that that pepsin can start catalyzing the reaction by which protein is broken down. The other thing that I wanted to say about the gastric phase, again, gastrin is a very important part of the regulation of the gastric phase. So what happens is gastrin, as it's secreted by the enteroendocrine cells, gets into the blood and circulates back to the stomach. And then, as I mentioned a second ago, gastrin stimulates the smooth muscles, so you get more forceful contractions of the stomach, and it also stimulates the parietal cells to increase the amount of hydrochloric acid that they are secreting. The other important thing that gastrin does is it helps to stimulate the contraction of the pyloric sphincter. So it helps to keep that pyloric sphincter contracted, and in doing so, that helps to essentially isolate what's happening in the stomach from the duodenum, from the small intestine, right? And that's so to give time for the intestinal phase to kick in. So the intestinal phase is initiated when chyme reaches the small intestine, right? So at this point, this pyloric sphincter opens a little bit, several milliliters of chyme are moved into the duodenum, and the intestinal reflex or the intestinal phase is initiated. So an important component of the intestinal phase involves what we call the intestinal reflex. So the intestinal reflex is going to be the reflex that opposes the cephalic and gastric reflexes, right? Both the cephalic and the gastric reflexes were kind of, especially the gastric, but are kind of pro-stomach phases, right? The cephalic phase preps the stomach for receiving food, and then the gastric phase involves those activities that are involved with the mechanical and chemical digestion of food and the preparation and or I should say the making of chyme. And then the intestinal reflex, what the intestinal reflex does is that, so it's initiated by chyme entering the duodenum. And when that occurs, signals are sent to the medulla oblongata. And what the medulla does in response to that is it decreases activity of the stomach, okay? So let me say that again. So when chyme is moved into the duodenum, that stimulates nerve signals, and those nerve signals, sensory nerve signals, are sent to the medulla oblongata. And in response to that, the activity within the stomach, the smooth muscle within the stomach, stops contracting as vigorously. So it slows down activity in the stomach. It also prompts the release of two very important hormones, and those hormones are cholecystokinin and secretin. So both cholecystokinin and secretin, which I described on Monday, I said, think of these as being pro-intestine hormones, right? And what I meant by that is these are the hormones that are responsible for regulating activity within the small intestine. And in order for, and again, this is all about coordination of what's occurring in the stomach and what's happening in the duodenum. So both CCK and secretin will increase activity in the duodenum, right? And I'll talk about specifically how here in just a moment. But then importantly, they both together have a synergistic effect, and that they decrease motility of the stomach. And that's really important because when the pyloric sphincter opens, and the duodenum moves chyme, and the stomach moves chyme into the duodenum, you now want activity in the duodenum to occur so that that chyme can be neutralized, and then also chemical digestion of the macromolecules found in the chyme can occur. And you don't want a bunch of activity in the stomach still taking place, because if it did, that would potentially, it would increase the likelihood that chyme would inadvertently move into the duodenum when the duodenum is not prepared to receive it, right? So CCK and secretin, again, think of them as pro-duodenum and kind of as being hormones that help to quiet down or calm the activities that take place in the stomach. Okay, so here is an image that comes from your textbook that just kind of summarizes these three different phases. And again, we'll talk more about CCK and secretin here in just a moment. So now we're going to move on, and we're going to talk And let me see how I'm doing on time. My hope is to get through. Okay, good. So my hope is to get through at least all the way through the lower GI tract, right? So we're going to be talking now about the duodenum, jejunum, and ileum, the three parts of the small intestine, the large intestine, and then a bit about the liver, the gallbladder, and the pancreas as far as what it is I want you to take away from that material. So the duodenum, again, is the first portion of the small intestine that is connected to the stomach. This is the portion of the small intestine that receives the chyme from the stomach. It's a relatively short segment. Attached to the duodenum is the jejunum, which, as I mentioned on Monday, is where the vast majority of nutrient absorption takes place and water reabsorption takes place. And then the ileum is the last portion of the small intestine that is characterized by the presence of Peyer's patches, which are those lymphatic groups of lymphatic, that diffuse lymphatic tissue that plays an important role in alerting the immune system if there are pathogens present in the stomach or the small intestinal contents. Okay, so just a little bit about the microscopic anatomy of the small intestine. So when you look at the small intestine, you'll notice that the mucosa and the submucosa are folded into what we call circular folds. These circular folds play an important role in helping to propel chyme through the lumen of the small intestine. You find many more circular folds in the duodenum and the jejunum than you do in the ileum. And then if we take a little bit closer look, or we pan in, we see that the mucosa itself is made up of what we call villi. Within the villi themselves, this is where we're going to, I'll talk about these in a moment, where we're going to find the enteroendocrine cells, which are the cells in the context of the small intestine that secrete cholecystokinin and secretin, goblet cells that secrete mucin, which becomes mucus. And then we have what are called unicellular gland cells, which make an important enzyme called enteropeptidase. And then you can see, so if you look over at this picture right here, you can see those villi, and you can see the cells I was just discussing. So again, goblet cells make mucus. Of course, we have simple columnar epithelial tissue that lines the lumen of the stomach, and as that type of tissue is very well suited for absorption. And that's because of the modifications in the plasma membrane of those cells, which are called the microvilli, which we'll talk about in a second. Goblet cells make mucus. These unicellular gland cells make enteropeptidase. And then we have the enteroendocrine cells, which secrete cholecystokinin and secretin. And then, as I was just touching on the surface of the simple columnar epithelial tissue, we have what are called microvilli. So microvilli are these modifications of the plasma membrane that increase the surface area that's available for absorption in the small intestine. And what we find in the microvilli are what we call brush border and brush border enzymes. So brush border enzymes also play a really important role in chemical digestion. We have brush border enzymes that catalyze the reactions by which carbohydrates are broken down. We have those that catalyze the reaction by which proteins are broken down, fats, and then also nucleic acids, which we won't focus as much on. Okay, this is just a picture that comes from your textbook. Just helps you appreciate what the circular folds look like, what the villi look like, and then you can't see, we're too far out to really be able to see the microvilli in this particular slide. So other things I want you to know about the duodenum. So specifically when we talk about CCK and secretin, and I mentioned this on Monday, but CCK and secretin work synergistically. And what CCK specifically does, so CCK, when the chyme that enters the duodenum has a high fat content, that stimulates the enteroendocrine cells to secrete CCK. CCK gets into the blood, it travels to the gallbladder, and it stimulates the gallbladder, and it causes the gallbladder to contract and to dump bile into the duodenum, right? And in bile, when it comes into contact with fat, emulsifies that fat, so breaks it up into droplets, so that the lipases, the pancreatic lipases, which are the enzymes that catalyze the reactions by which fats are broken down, have a greater surface area to work on, right? Secretin what's secretin, or you get an increase in secretin when the enteroendocrine cells that secrete secretin are exposed to a low pH. So chyme itself has a low pH, right? It's got hydrochloric acid in it. So what happens is when those enteroendocrine cells are exposed to low pH environments, like what you find with chyme, they secrete secretin. Secretin gets into the blood, it travels to the pancreas, and it causes the pancreas to make and secrete bicarbonate and to dump bicarb into the duodenum. And what that does is it neutralizes that very acidic chyme. So CCK and secretin together play a really important role in the digestions of fat, the digestion of proteins. Both of these, and they're both stimulating to the pancreas and increase the amount of enzymes that the pancreas makes and dumps into the duodenum. And those enzymes are going to be responsible for chemically digesting all of the macromolecules. So fats, carbs, proteins, and also nucleic acids. Although I should say, let me back up a moment. The vast majority again of, so we have carbohydrate digestion that begins in the mouth and then continues in the small intestine. And the brush border enzymes, those on the microvilli, also play a very, very important role in carbohydrate digestion. So I'll say that now, and I'll say it again when we get there. Let me see if there's anything else I want to say from that. Oh, geez, I can't believe I left out trypsin. So the other important thing that happens within the duodenum. So again, the duodenum receives the contents, and you can see I think it's pretty, this is a great picture to help you visualize it. So the duodenum receives secretions from the gallbladder, which again would be bile, and then from the pancreas. And the pancreas, the secretions of the pancreas, as hopefully we'll talk about here in just a second, are going to include enzymes that break down the macromolecules. So lipases, amylases, proteases, and then the proteases specifically are going to be an enzyme called trypsin. So trypsin is the activated enzyme that is activated from trypsinogen. Trypsinogen gets activated to trypsin by enteropeptidase, right? Which we talked about earlier when we were going over the different cell types that we find. And again, we'll talk about it more in a moment when I get to that slide. Okay, and then the jejunum and the ileum, I think I said most of what I want to say about those portions of the small intestine already. And that same information is here on that slide. Okay, when it comes to movement in the small intestine, so segmentation, actually, let me start with peristalsis. So peristalsis, we've already talked about, right? Peristalsis occurs during the intestinal phase when we were talking about the cephalic, gastric, and intestinal phase. Peristalsis itself is, again, that wave-like, that inchworm wave-like movement that is unidirectional. There's something called motilin, which is a chemical released by the duodenum that helps to stimulate peristalsis within the duodenum. And pardon me. And then we also have a type of movement called segmentation. So segmentation is different from peristalsis. Well, it's the same as peristalsis in that it is movement, forward movement, right? However, it's different from peristalsis in the sense that its segmentation is kind of like if peristalsis and got together with mixing, right? So it has elements of both. It has elements of mixing and elements of peristalsis. The long and short reflexes that we discussed on Monday, they play a really important role in controlling segmentation. So, and again, these are when we talk about various afflictions, homeostatic imbalances that are related to the alimentary canal. Irritable bowel syndrome is one of them. And irritable bowel syndrome can be, it's this dysregulation, in part at least, a dysregulation of movement, not just at the large intestine, but also can occur within the small intestine as well. Okay. And then one last reflex I want to discuss. So we have what's called the gastroileal reflex. So this is the reflex that essentially allows, it's no longer called chyme, but the contents of the small intestine, after they have made their way through the ileum, those contents are going to move into the first portion of the large intestine, which is called the cecum. So there is a sphincter that controls the movement of substances from the ileum to the cecum, and that sphincter is called the ileocecal sphincter. And so the gastroileal reflex is the reflex that essentially is responsible for contraction of the ileum, which increases pressure and opens up the ileocecal sphincter and moves the items within the small intestine into the large intestine. CCK, cholecystokinin, also helps with this. So again, cholecystokinin is a hormone. It's in the blood. So when that blood travels to the ileum between and the location where you find that ileocecal sphincter, geez, tongue twisters, that causes that sphincter to relax, which makes it makes the movement of substances from the ileum to the cecum a little bit more fluid. Okay, so intestinal juice. When we look at what we call intestinal crypts. Intestinal crypts that's the name that we give to that. Essentially, it's another name for a portion of the villi. And what we find with that term intestinal juice is used to describe the secretions of these different cells that we discussed, right? So intestinal juice itself tends to be slightly alkaline because of the presence of bicarb. The juice, if you will, itself is mostly isotonic with the blood plasma. So it's made up largely of water. Of course, it has mucus because we have secretions from the goblet cells. Also within the intestinal juice, you're going to find these secretions from these unicellular gland cells, which include enteropeptidase, right? So again, enteropeptidase is the enzyme that catalyzes the conversion of trypsinogen to trypsin. Okay, so that's really important. So make sure you know that. And is there anything else I want you to know? And then here you can see the enteroendocrine cells, which again secrete secretin and cholecystokinin.
Hey, Professor Hanley, I have a question really fast.
Yes.
Where does the bicarb come from that you're talking about?
Yes, the bicarb comes from the pancreas. So if we go back to this slide right here, I'm not because it's covered in lab. I'm not talking in lecture about the ducts that that connect the accessory digestive organs to the duodenum. Just suffice it to say that there are ducts that connect the gallbladder and the liver and the pancreas, and those ducts all dump into, by connect, I mean they connect those structures to the duodenum. So the substances that are made by the pancreas are dumped into the duodenum. So the substances from the pancreas are going to include bicarb, and then all of the enzymes used to digest the macromolecules or the precursors for them. So in the case of bicarb, to answer your question, bicarb is made by the pancreas, dumped into the duodenum. Trypsinogen is made by the pancreas, dumped into the duodenum, and then converted to trypsin by enteropeptidase, which is secreted by the cells of the duodenum, by those unicellular gland cells.
Thank you.
Oh yeah, no problem. So let me see what I want to say. Kind of running low on time. So I'm not going to spend any time on this, except that don't use the expression stomach flu. It's totally misleading. Flu, that term, the flu, is used to describe, and by and large, it's a reference to an upper respiratory tract infection, right, by the influenza virus. And what people call the stomach flu is somehow an irritation of the GI system. Now, that's not to say, for what it's worth, there are viruses that can cause upper respiratory tract types of symptoms. Can cause runny nose, can cause fever, can cause a cough, and also are irritating to the GI system. It just so happens that the influenza virus is not one of them, by and large. So the stomach, most of the time, if you are sick to your stomach, if you're nauseated, it's either because you have food I'm talking about things that are pathogenic in nature. Um, when it comes to pathogens, it could be because it's rarely, but can be because of food poisoning. And also there are a variety of different viruses out there that are irritating to both viruses and I should say other types of parasites that are irritating to the alimentary canal and cause nausea and vomiting. I'm not going to talk about typhoid fever or cholera. They're very interesting, but I just want to get through a couple more slides. I specifically want to get to the pancreas before we're done. So here's what I want you to know about the liver. You can use these slides to study with for the lab exam too. So the liver itself is made up of 4 partially separated lobes, right, and 2 ligaments. So those 4 partially separated lobes is because we have a right and a left. Then if you look at the back side, we also have some additional lobes. So we have the caudate lobe, geez, if I could talk, and then the quadrate lobe, right? I'm not going to ask you that much anatomical detail in lecture, so I'm going to skip over this. The liver is very important. The liver, in the context of the digestive system, of course, makes and secretes bile and then moves that bile to the gallbladder so that the gallbladder can store and secrete bile. If you look at liver tissue itself, it's broken into what we call lobules. And then at the corner of each lobule, we have what are called portal triads. So the portal triads are made up of an artery and a vein and then a bile duct. And that bile duct, again, is there to take the bile from the liver to the gallbladder. And then it also takes it in part to the duodenum. And if the gallbladder has to be removed, the liver can compensate for the gallbladder and still make enough bile to emulsify fats. Okay, so bile, again, made by the liver, stored and secreted by the gallbladder. Bile itself is made up of water, bile salts, bicarb ions, various pigments, mucus, and what bile does is it emulsifies fat. Hepatic portal circulation, everybody should be familiar with because we talked about it when we were doing the circulatory system, right? But just by way of reminder, portal circulation is where you have 2 connected capillary beds. The first is responsible for gas exchange, like what we see at a typical capillary bed. And then the second serves the purpose of exchange of substances that are not gases. So in the context of hepatic portal circulation, this describes the circulation between the small intestine and the liver. So you have blood, nutrient-rich blood that is drained from the small intestine is then run through a second capillary bed in the liver. And then what happens is exchange of those nutrients between the liver and the blood. And what the liver does is it modifies substance that might otherwise be caustic. So it helps to neutralize free radicals. Sometimes it can take innocuous substances and inadvertently make those reactive, right? The phenomena by which substances are substances that you take by mouth are altered by the liver is called the first pass effect. First pass effect plays an absolutely critical role in pharmacology. Oftentimes, what you ingest, if you swallow a pill, is not what the cells of your body are seeing, right? Supplements are a good example of this. Glutathione, for example, which is a very potent antioxidant. If you go to a GNC or any supplement store and they are selling you glutathione pills, it's a total ruse. Glutathione, if you take it by mouth, because of first pass effect, it renders glutathione completely inactive. If you want to increase your glutathione levels, what you really should take is something called N-acetylcysteine or NAC. And NAC, when it goes through the liver, is converted into glutathione. So anyway, that's just kind of an interesting side note. Oh man. Okay, you guys. So I'm mostly out of time. I think I said everything I want to say in here as far as I do expect you to know CCK causes the gallbladder to contract and release bile. And then for the pancreas, I go through the pancreas slides. I said most of what is on them. And I do expect you to know what the pancreas makes and secretes into the duodenum. And so again, and then I have here trypsinogen, and I bolded it to remind everybody this is really important. It's activated to trypsin by enteropeptidase. This is the enzyme that is made and secreted by those unicellular gland cells that we find in the intestinal crypts, right? In the intestinal, in the folds, in the villi. So, and then make sure you go through this slide. I said everything that's on here, but go through. And then that's where we're going to end. So that's going to be it for the exam. We'll end with everything up through the large intestine, unless go through the rest of the slides. If there is content that I verbally covered earlier on, but that's in here, make sure you go through and read it. You are responsible for it. Anything I said by mouth, anything I said out of my mouth, anything that comes out of my mouth, I suppose, is subject for testing, right? So go through, I'm just kind of running through this to see if there's anything. I don't think, but I'm not going to ask you any specifics that I didn't, and I didn't get to any of that. Didn't get to any of that. Yeah, and then there is some information in the chemical digestion portion of the PowerPoint that we did talk about. So make sure you go through these, because I did talk about this. I just talked about it in slides that were relevant to this, but just earlier on, right? So know the different enzymes that we have that are responsible for breaking down the macromolecules, right? Amylases for carbohydrates and the brush border enzymes that are present. These are more brush border enzymes. Don't worry about that. Proteins, right? We have proteases. So we have pepsin in the stomach. We have trypsin in the small intestine, but trypsin is, and we have pepsin is made by pepsinogen being converted to pepsin by hydrochloric acid. We have trypsinogen to trypsin by enteropeptidase, right? But those are the enzymes that are responsible for breaking down proteins. Again, hydrochloric acid plays a critical role in the production of pepsin. So it's very important in protein digestion. And then we talked about trypsinogen. We didn't talk about chymotrypsinogen, but these are just other associated protein digesting enzymes, which are called proteases. Enteropeptidase we did talk about. So go through these slides of digestion, because we did talk about these. And then the lipases we talked about also. These are responsible for digesting fats. Fats are definitely best digested after they are emulsified by bile. But I'm not going to ask you specifically about lipid absorption because I didn't get a chance to get to it. And nor am I going to ask you about nucleic acid. Yeah, and I won't ask you about those. Okay.